So today we are going to discuss about the germplasm conservation. So in case of germplasm conservation, before discussing the germplasm conservation, we have learned about the how our plant biotechnology lab should be. What are the criteria which will be fulfilled so that our lab will be functional for tissue culture technology. So then we were able to learn what are the different medias, commercially available medias, which are used for different plants to convert those plants into the tissue culture technology. Then later on, we were able to understand if we will take the explant from the natural source, then we was able to grow it under the lab conditions to form the callus. Then from callus, we were able to understand how suspension culture is made. We also learned about the protoplast, isolation, fusion, and differentiation, and regeneration of protoplast. So, to maintain all the valuable things which we did in our previous lecture, or we learned that we are having hybrid plants. Now we have to maintain the hybrid plant for future use. So, for that purpose, we have to go for germplasm conservation. And this germplasm conservation was initially started for those plant species which are endangered, or there are some commercially available species for which we need to go for the germplasm conservation. So, germplasm conservation is the most useful and successful technique where we are able to store our live information of genes present in the plant and can be conserved for longer period of time. And whenever we are in need of these plants or these sources, we can take and regenerate it whenever we are having its requirement. So this germplasm also provides the raw material for the plant breeders. So anytime or any breeding program when we have to start, we can take the germplasm, then follow our breeding technology or the breeding programs we have to follow. Earlier, traditionally, we can say we used to store, we used to go for conservation, but in the form of seeds, tubers, or bulbs, roots, rhizomes, buds, or cutting of the plants, or under natural conditions, we used to keep field plot for our conservation, or nurseries, plant nurseries, or botanical gardens nowadays we are having available, and greenhouse effect. So collectively, we can call these as natural gene banks. Natural gene banks. But still, we are having difficulties by applying such mode of conservations. We are having some plant species whose seed are not fully developed, or there is early embryo regeneration. So these types of plants can't be maintained in nurseries or the greenhouses, and the cost of or the cost in maintaining these nurseries or greenhouse are very expensive, and sometimes we will be in a we will be in a loss if we will keep our natural source under natural conditions. Like under natural conditions, these plants will be vulnerable to insects, they will get attacked by the pathogens, and there will be various climatical hazards which will affect our natural germplasm conservation. Till now, it has been estimated that 20,000 or above species are in endangered list. For those plants, to maintain these plants, we maintain these plants under plant tissue culture technology. We take these plants, grow it under the lab so that there will be again they will increase. We will be able to increase their number under natural conditions, and also we can maintain these endangered species plant for germplasm conservation under the low temperature conditions as well. but still these are not effective for the longer time of conservations. So the germplasm conservation are having certain significances as well. First, the genetic diversity can be preserved for longer period of time, and whenever we are in need of this diversity or we are in need of this plant, we can take its germplasm, then we can be make it available to the whosoever wants it, and we can use this germplasm conservation for endangered plants. We can also use this germplasm conservation for those seeds which are short-lived or recalcitrant. They don't grow on time or they are having problem while germination. Even we can use this germplasm conservation for canola crop seeds, as these clones are having heterogeneity in their genome, so it will be beneficial for us to store these clonal crop seeds as well. So there is the one worldwide we have they have established one seed bank. That seed bank is called as International Board of Plant Genetic Resources. International Board of Plant Genetic Resources, IBPRG, which was established to store the germplasm. So they what they do, they collect the germplasm of the material, then conserve it. Then whenever they are in need, they utilize this germplasm conservation. Worldwide, they are able to produce its germplasm. Whenever if we are having any experimental research, we don't have that species in our Kashmir. So what we will do, we can order our germplasm from there, then we can start work here. So broadly, on broader categories, we are having two approaches for germplasm conservation. First is in situ conservation, and second is the ex situ conservation. So we have to give more emphasis on the ex situ conservation. It has been, or it is one of the best method for conservation of germplasm from different plants. It is the best method. What we do, we take material in the form of seeds, or we can take material in from the in vitro cultures, like we take callus, cells, organs, then we preserve it, and the whole collection of this preservation we can call it as gene bank. In gene bank we are able to store our material under longer terms, means for longer period of time, with suitable condition. Before starting our this conservation process, we should be having adequate knowledge that how and what are the different techniques which are involved in the preservation of these samples. Like first we should be able which plants material we can use as a germplasm conservation, means we should be able to have adequate knowledge about the sampling, because every time we can't take only shoot tip cultures, so we have to take different samples from different plant species. We should be having adequate knowledge for that. Then we should be able to regeneration process when we store this under the in vitro, under cryopreservation or any other preservation, then we should be able to know how to regenerate these plants, or how to regenerate these materials under lab conditions. And the most important is its maintenance, as we are storing our material for longer period of time. So, in vitro preservation of germplasm are basically divided into four different approaches. We can store our germplasm via the cryopreservation first. Second, we can store minimum growth storage, means we will be able to allow them to grow but at a minimum growth. So third will we can keep our plant for preservation under cold storage condition. And last one is the we can keep our plants or germplasm under low pressure and low oxygen storage. So we will discuss first about the cryopreservation technique. Under the conventional method, these germplasm are prone to get attacked by different bacteria, fungi, means different pests and pathogens. So means conventional method is not beneficial. Or this conventional method will be able to get or will be able to have climatical effect, means climatic disaster will be there, or natural disaster. Sometimes there is increase in temperature, that will be climatic, or natural disaster will come under the flood. If there is a flood, that will go for the natural disaster. And sometimes it has been said that this conversion method is also getting affected by political and economical causes. Like in Kashmir, we are having forests, but due to these politicians, some politicians, these forests are cut down and they sell these timbers. So it means our natural resources are also affected by politicians as well for the economic cause. Even we are, so we will, if we will talk about the storage of the seeds under low conditions or under the low storage conditions, some we are having some seeds which are having whose viability is very short. Like we are having examples of cocoa, jackfruit, bread, breadfruit, avocado, or coconut. All these include their seed is viable only for shorter period of time. So in this case, we have to think something different. Or there are some plant species which only propagate by vegetative propagation, means they are not able to form their seeds. So what we will do in that case, we will take seed or its tubers or cutting, then we will go for its preservation. But preservation of such type of material will be laborious, cost effective, and we will be need of larger space to store these things. Best storage is in the form of seeds or in the form of shoot tip, root tip cultures, means meristematic cells we can store. Or we are having certain plants which are only for shorter period of time they are active, like we are having banana trees, yeah, potato, cassava, they only remain active for shorter period of time. There are also many other species for which we were not able to go for efficient reproductive methods, means till now we were not able to reproduce them effectively. These trees include banana, coconut, oil palm, or date palm. So, for all with all these factors, they realized that the cryopreservation is the best technology for the germplasm conservation. So, even if we will think that we will culture, what we do, we take these whatever we discussed, all the seeds or all the species, we take these material into the lab and then go for tissue culture technology. But this tissue culture technology has its own disadvantages as well. As we know, while subculturing of material again and again, sometimes there is the loss of some genes or there is the somoclonal variations. If we will store our germplasm in the form of in vitro cultures, that also is not beneficial for us, because if we will take explant then again and again we reculture it, means culturing करेंगे subculturing करेंगे, that time also we will be able to get some somoclonal variations. It means that in tissue culture technology we will not be able to store our germplasm in a wild state conditions. So what we do, we simply take our material, then we will allow it to, then we will take our material, then place our materials in the small vials, then we will take these vials into the liquid nitrogen and whose temperature is minus 196 degree centigrade. So the important steps is in the cryopreservation is the pre-treatment. We can't directly take our sample then place in the vial and place in the liquid nitrogen, no. So we have to be sure certain protocols, certain steps are there which we have to initiate for cryopreservation. So we have to go for, it means we have to go for pre-treatment. We have to go for pre-treatment if we are taking our explant from plants. For example, we are taking tropical plants. We can't directly cut tip of the tropical plant, then place it into vial, then in the minus liquid nitrogen. No. So what we do here is we take a plant, we allow it to acclimatize under cold conditions. If we are taking plant from the tropical region, we acclimatize it under the cold conditions. So how? Because these are the warm temperature plants, we give them short warm days and long colder days so that this plant will be able to grow under this condition. Means we will change its environmental conditions, but we have to do this change in such a way that this plant should be able to survive and grow. If the growth is the minimum, that will work out, but this plant should be able to survive under this condition. When we realize that these plants are growing, then we can take this explant, then we will immerse these explant into the osmotic medium. We will place this explant into the osmotic medium. So the osmotic medium which we are using in case of cryopreservation is also called as cryoprotectants. And the best example of the cryoprotectant is the DMSO, dimethyl sulfoxide. We have to use DMSO as a cryoprotectant in the concentration of 5%. We can take our explant and place it in 5% DMSO for 2 days. So for 2 days, this is the maximum time we can take this our explant into the cryoprotectant for 2 days, but it is not applicable to all plants. There are some plant species which we have to expose to cryoprotectant only for 30 minutes or as long as for 2 hours. Then we have to, but we have to be sure that our explant should not get damaged or our while our exposing it to the cryoprotectant. मीन्स हमें एक ऑप्टिमम रेंज चेक देखनी है कि हमारे प्लांट स्पीशीज को कितनी देर हम लोग क्रायो प्रोटेक्टेंट में रख सकते हैं। उसका ये है कि अगर हमने ओवर टाइम दे दिया उसमें क्रायो प्रोटेक्ट में, दैट विल डैमेज आवर टिश्यूज, क्योंकि उसकी ऑस्मेलिट ये मोलैरिटी क्या है, हाई है, दिस इज 5%। सो डिहाइड्रेशन इतनी ज़्यादा हो जाएगी दैट विल डैमेज द इंटरनल ऑर्गन्स ऑफ द सेल। सो we have to be very specific for exposure. So it means we are done with the optimization of cryoprotector. Then slowly under control condition we will increase our cooling temperature. Then we will increase our cooling temperature. Then finally we will immerse our vials into the liquid nitrogen. So this was the how we give the pre-treatment for cryopreservation technique. Now we have to go for another is the when we have to take the samples back for regeneration or rewarming or thawing process. This thawing process is one of the critical step which is involved in the cryopreservation. Now our sample is in minus 196 degrees centigrade. If we will take our sample and allow it to cool at room temperature, as there is a vast drastic change and this will take time to come into to get adjusted with the environment, there is probability that there will be the more damage to the sample. So what scientists have learned, they learned that while taking vials from minus 196 degrees centigrade, they directly expose these vials to the hot water. The temperature of that hot water is 40 degrees centigrade, and they keep these vials under 40 degree centigrade for 1 to 2 minutes. That means directly cooling से हमने क्या किया, directly hot water में डाला, because we are having protected cryoprotectants over our sample, so it will not damage our sample. So then from after 1 to 2 minutes is over, then we take our vials, then place it in the cold water whose temperature is 25 degrees centigrade. We have all the procedure which we are using in the thawing process should be very rapid. Otherwise, if we will take little time, the cryoprotectant will crystallize while coming in contact with the environment. It will get crystallized क्योंकि इसको टाइम मिल रहा है ना फंक्शनिंग के लिए. We are not directly allowing it to go for हॉट वाटर में नहीं दे रहे इसको. स्लोली आहिस्ता आहिस्ता इसका टेम्परेचर क्या हो रहा है, वार्म हो रहा है. मीन्स -196, 180, 160, 150. Then the cryoprotect in this process, the solutions may crystallize, and this crystallization will lead to the death of our explant. Will lead to our death of the explant. So we have now our explant in our hand by thawing process. Sometimes we can go for washing of the cryoprotectant from these explants, but in many cases they have observed that we will not they don't decant this cryoprotectant, they keep it on the explants, and they have observed that keeping these cryoprotectant on the explant and went for the growth, that is the differentiation, they observed that there is the better growth. हमने सैंपल लिया, अब हमने क्या इसकी thawing process की. Now we have to take sample from these vials. Now we have to allow it to the differentiation process. So in many cases they have observed कि हम लोग इसमें cryoprotectant को wash नहीं कर रहे हैं. इस cryoprotectant को हम लोग explant के surface पे रहने देते हैं. They have observed, in many cases they have observed the better growth. And in many cases they have observed कि नहीं, first we have to go for the washing, rinsing of this cryoprotectant, then only plants will be able to go for the differentiation process. So it depends on your plant species. So when we allow our plant species or our explant to go for the differentiation process, we always keep our explant under low or no light condition for first week. As we know, when there is a presence of light, the phenolic compounds will get released from these samples, and they won't allow to form the callus under in vitro conditions. So we have to follow standard growth conditions. It can be also possible that we have to take two different media. First is the recovery media, when the explant is directly taken from the cryoprotectant. That time we will make its one media which will act as a recovery media. When we realize that the material which is there has recovered properly, then we will place this explant in the multiplication media. In sometimes we can in multiplication media we add auxins so that there is the easily formation of calluses. So this was a basic intro for the cryopreservation. Again, we are having different techniques which are involved in the cryopreservation technique. So first technique is the stepwise cooling. First is the stepwise cooling, or we can say it slow cooling, controlled rate cooling as well. This type of stepwise type of cooling is mostly used for the wood plants. Mostly used for the wood plants. What we do in case of wood plants, we take sample in the midwinters. We take sample from these wood plants in the midwinters. Then again, while taking these explants from the natural source, allowing it to allowing it in a in vitro conditions, again we are giving additional chilling. We are giving additional chilling means we are again allowing it under harsh cold conditions, due to which there is a dehydration. Then we will place this into cryoprotectant, then from cryoprotectant into the liquid nitrogen. But there is a stepwise process. There is a stepwise process. Like we have take our explant, we have treated with this explant with the cryoprotectant, and we allow it to freeze at less than 1 degree centigrade. Means from we will start its freezing from 0 degree. So we kept this 0 degree for 1 minute. Then we again increase little, decrease our temperature till we reach minus 40 degrees centigrade. We have to gradually decrease our temperature, means cooling temperature. When we reach it up to minus 40 degrees centigrade, then directly from this minus 40 we will place our sample in the minus 196 degrees centigrade. This is how stepwise cooling process is carried. Another is the rapid cooling or solution based vitrification. What we do here is, as name indicates, rapid cooling. We take our sample, place it in the cryoprotectant, then from cryoprotectant we directly place it into the liquid nitrogen. We don't give any pre-treatment. Pre-treatment is only cryoprotectant. Rest we don't go for slow processing. We directly keep these plants under in vitro conditions. The third is the encapsulation and dehydration. Encapsulation means you have to encapsulate and dehydrate the sample, as name itself said. Encapsulation and dehydration process. In this process, what we do, we take our explant, encapsulate it. We can use agarose for encapsulation, as we use in case of immobilized cell, or we can use alginate. Then we go for dehydration process for 20 hours of this explant. Then we allow it to dry it under the silica gel. So when there is a complete dehydration process, then only we will place our explant in the liquid nitrogen. So this was the third technique. Another is simply encapsulation vitrification. Encapsulate our explant, then we will place it into the cryoprotector, then directly into the liquid nitrogen, means there is no dehydration process. And the simple and last is the desiccation process. We take our seeds in a dry form. If these seeds are not dry, then we allow it to dry over the silica gel. You might have seen silica gels in your shoes anywhere, because they help in the drying the things. They absorb the environmental moisture. They keep things fungal free or moisture free. So for these seeds, we should be keeping moisture content within in the range of 3% to 7%. Then we can take these seeds, then place it into the cryoprotection, then in the liquid nitrogen. Or we can use embryonic cells as well, for whose we can for those we can take water content, or we can dry it up to the 7% or 20% water content, depends on their weight. Then these embryonic cells are placed in the cryoprotectant, then into the liquid nitrogen. So this was how different techniques are involved in the cryoprotectant. Now the last point for today is, we are having different cryoprotectants. Scientists have made different cryoprotectants which are applicable for different samples along with their different techniques. Like we are familiar with the DMSO. This DMSO is composed of 5 to 10% DMSO, dimethyl sulfoxide. This DMSO can be used for, as a cryoprotectant, can be used for embryonic cultures of conifers or mosses, algae, and in this case they are using stepwise cryopreservation technique. They are using stepwise cryopreservation technique. We can use sucrose as a cryoprotectant with the concentration of 0.75 molar. So the technique here for sucrose we are using is the encapsulate and dehydration process. So the samples which can be used for sucrose are shoot tips, somatic embryos, ferns or mosses. So we are having different techniques for cryopreservation. We are having different cryoprotectants which are having different compositions. Like we are having another, we can use DMSO along with the sucrose as a cryoprotectant. As name indicates, DMSO भी होगा और sucrose भी होगा. So there we are using 10% DMSO along with the 0.6 molar sucrose. This type of cryoprotectant is used for embryonic cultures. We are using this for the embryonic cultures, and there we are giving them the stepwise cooling. We are giving them the stepwise cooling. Mostly it has been observed that stepwise cooling procedure is the best procedure for cryopreservation techniques. And the last cryoprotectant is DGS. In this case, what they are using 1 molar DMSO, 1 molar glycerol, and 2 molar sucrose. So the samples which are used for this cryoprotectant are differentiated cultures. Differentiated cultures, and the technique is the same stepwise technique we have to apply so that our samples will not get affected.